I am delighted to present DNTOX to you. We are a Düsseldorf-based startup company, and I'm going to speak about our mission, chemical safety without using animals for testing and to enable it for the industry. It is generally known that chemicals can cause permanent damage to the human brain. And such an exposure can happen because of contamination in the air, in water, pesticides, and softeners. And the developing human brain is particularly sensitive to such environmental influences. And if during the brain development there is such a damage, we talk about developmental neurotoxicity, DNT. So this means that from now on, if I speak about DNT, this is what it means, developmental neurotoxicity. And DNT has different implications. For instance, ADHD, autism, learning deficits, but also IQ losses. And in general, a central problem is that only approximately 200 substances, chemicals among them, have been tested for their potential to do harm to the human brain in its development stage. And this considerable gap of available data is due to the fact that DNT testing is not stipulated by law. They are only carried out if there are hints to adult neurotoxicity. And these tests are done with animal experiments according to the regulatory standard. And the animal experiments are not suited for comprehensive DNT testing. To carry out such tests, you need a large number of animals. This is just for DNT testing, the numbers you see here, and they are very cost and time intensive. And of course, as the previous speakers also mentioned, there is the problem of the adaptability or the applicability to the human being. And because of that, there is a big need of having alternative methods to test the large number of chemicals there is no information about at this point yet. And in recent years, there have been many attempts coming from different directions to have human-based in vitro methods for such tests and to establish them. There were corporations, regulatory authorities were involved, the OECD, OECD, for instance, or the EFSA, European Food Safety Agency, and researchers, Food Safety Authority rather, and people got together to develop a roadmap which makes the alternative method and their use for regulatory use to make this possible, particularly when it comes to the approval and reapproval of pesticides. Crop protection agents. And one of the most important parties involved and person involved is Ellen Fritsche, our co-founder and managing director. And it was based on her activity in the working group that the DNTOX was founded in 2022 in order to enable DNT in vitro testing, particularly make this be accessible for the industry and regulatory authorities. If there is no application, there can be no change of the legal prerequisites. This brings me to our human-based test methods. How are we doing this and how does it work? In the first place, we use human-based cell systems that represent different developments of the brain in vitro. And our big advantage for the industry because we are more cost-effective and we're faster. We can deliver data within 6 weeks. And with that, we can also increase the relevance of our results for human safety, and particularly because we are moving within the realm of the same species. Modeling the brain development is done. The process is quite complex of the brain development, and we can depict it with our processes. we use human neuronal precursor cells that we cultivate in 3D cell aggregates, spheroids, no organoids, as we heard in the previous presentations. And these spheroids, we call them neurospheres. And these neurospheres can be used for the proliferation of, and can analyze the neuronal precursor cells, the migration, the differentiation in different effector cell types of the human brain, in neurons, astrocytes, and oligodendrocytes. But we also have other test methods that are based on human-induced iPCS cells, and the iPCS cells are used to analyze the formation and development of neural networks. This is a more complex process during the development. And of course, the very complex development of the brain cannot only be analyzed in one assay or one test. And this is why there was a cooperation by the Leibniz Research Institute for Environmental Medicine, the founding lab or the founding group of DNTOX, plus Konstanz University and the US American Environmental Authority. And these three laboratories developed test methods that were merged in the DNT test battery. And this merger of test methods created 17 endpoints of the human brain in different test methods that were created. And this is something that was also supported by OECD and EFSA in recent years. Thanks to these test methods, we are offering a service for the chemical and pharmaceutical industry. This means that the customers send us their substance candidates, and we expose the human cell systems in different test concentrations in our laboratories. And then we can apply different methods I'm going to speak about in more detail in a moment. We can use these methods to show how the different cell types or processes develop under the influence of the test substance, and how they change. Here you see a neurosphere which differentiates and migrates, and the processes are very normal here. On the right-hand side, you see that the differentiation of the cells is impaired by a DNT substance, for instance. But of course, we don't want to only make these effects visible, we also want to quantify them and translate them into data. And to this end, we have a complex and AI-based workflow that we developed. On the one hand, it carries out the quantification of different cell types, enables it, and at the same time, thanks to different classification models, also gives an answer to the crucial question if a chemical poses a danger to the human brain. And as we've just mentioned, we work with human neurospheres. They proliferate over some weeks, and we can grow them for some time, but they also migrate. They migrate to radially out of this sphere scan. You can see that here. And when they're exposed to DNT chemical, you see that there's a clear difference and that there is the inhibition of the process. Here you see the images I've just shown, the differentiation of the different cell types that have been immunostained, and we can make all these effects visible. Now, this is my favorite assay. This is the human neural network formation assay. And for this assay, we use standardized IPS-based culture models, different cell types that we apply on different multi-well plates, special plates, and they are equipped with electrodes at the bottom. And this enables us to analyze the inference of electrical signals that are sent back and forth in the system and to translate them into data. In the videos on the left-hand side, we see the different lights. This is the electrical activity of neural networks. The stronger the light, the more electrostatic the network is. On the right-hand side, you see a strongly impaired grid, which is no longer that active. Now we have to wait for a little while until we can move on to the next videos because because of the videos. But at DNTOX, we don't only work in the lab. Particularly our bioinformatic experts deal with the assessment and the evaluation of the huge amount of data that we generate. And as we said before, we test the substances in different concentrations. For instance, a nanomolar or micromolar, that is what we're talking about here. And we can generate the concentration efficacy curves and concentration response curves. And we summarize the values generated in the lab, and our mathematical models calculate this curve that matches the data points. What we also do is we also determine the concentration, which you can see at the bottom, where a cutoff or threshold was passed. And this is the line, this line at 65 or 70%, more or less. And this is what we call the benchmark concentration, BMC. This is a very important value that you also know from animal testing, and so as to give you an idea. And as mentioned before, the development of these testing methods is supported by regulatory authorities. In 2020, we, together with EFSA, we provided the proof of concept. And based on the performance of these tests, EFSA recommends the obligatory use of DNT test methods for pesticides in Europe. And the latest milestone on this path to a paradigm shift in the DNT sphere was achieved by the publication last year of an initial recommendation by OECD. And this document, published by the OECD, there are recommendations on the evaluation of the data that are generated with the DNT in vitro battery. And of course, we are very curious to see where this journey will take us and what will happen in order to get away from animal testing and to use in vitro testing methods. This proof of concept, I'd like to show you some data. It comprises the testing of different positive and negative substances, which were already tested in DNT animal experiments. And our testing methods identified all the negative substances as negative, and out of the positive substances, 68% more or less, depending on what you're looking at, could be identified in the in vitro model, and the rest was false negative. But we also know that there are some gaps in our test methods, and these explain the false negative results. This is the team of DNTOX. At this point, we work with 11 people. We're working on this highly exciting topic, and we all come from many different areas. And this is also interesting to see who works in these biotech startups and in these companies. Our managing director, that's Ellen. She's a doctor and toxicologist. Silke, she is responsible for finance and administration. And then we have other biologists and toxicologists, and we have 3 bioinformatics experts and mathematicians, and 3 technical assistants. And of course, our dog, Sherry, our office dog, she is very important too. So there's a lot of expertise on the team, and it's by no means limited to life sciences. And before concluding my presentation and before starting the breakout rooms, I would like to speak about the foundation of DNTOX. As I mentioned, we are a spin-off of a working group of a Leibniz Institute based in Düsseldorf. And when I did my master's thesis and my PhD thesis, and I completed my PhD thesis with Ellen in 2022, but prior to that, we already prepared everything to found our company. And I have to say that creating a startup at the beginning of my studies and my PhD, well, that wasn't necessarily what I had on my personal to-do list. And this is why I love this picture. I didn't choose the startup life chose me. And if you want to know more about setting up a startup company and the transfer of knowledge in the industry, please, I would be pleased to see you in the breakout rooms so that we can have a chat about this topic, and I'm looking forward to it. So I'd like to thank you very much for your attention and hope that you liked it. Thank you. Thank you. Great. We are still well on the way time-wise. I have two questions for you. The first one is, in cosmetics, animal testing is also carried out still. The service you're offering, can you also offer it for cosmetic products? Yes, we also have customers from the cosmetics industry. But the problem there is that animal experiments are forbidden anyway, unless the product contains chemicals that are subject to the European Chemicals Act, REACH. And REACH chemicals need to be subjected to animal testing. And this is why cosmetics companies also do testing with us. This also happened in the recent past, but it's less than the chemical and the pharmaceutical industry. I have another question. The iPSCs, the human cells, where do you get them from? A cell bank or from clinics? These are commercially viable cells from cell banks, and we buy them. It's not patient cells, and we induce them to become neural precursor cells, for instance. But there's also the possibility to use patient iPSCs, and we did that in recent years as well. So, in order to develop disease models based on iPSCs.